It's delightful to have all of you here. You've endured, I'm not sure what, to get here, but we're delighted to have so many of you here. I see a lot of familiar faces out there. For all of those of you who have come to one of these programs before, welcome back. Those of you who are here for the first time, we're glad to have you with us. These programs that the Koch Institute has initiated are an expression of our intent to bring sort of what's going on at MIT out to the community in as broad and as forceful way as we can. We hope you'll find this evening quite inspiring, and I hope you'll look at your program and consider coming to some of the other events that will be sprinkled essentially 4 times across the calendar year. Tonight's program is focused on the tools and devices that are emerging in the oncology clinics in the marketplace. These With Insight solution lectures are breaking into essentially 3 chapters. We try to start in the beginning by someone who's really at the throttle of the problem. In this case, a physician who has been doing the management of this disease throughout her career, and she'll give us her view of it. Then we move into MIT laboratories, and we try to learn what it is that are examples of new technologies in a particular topic. In this case, we'll have Michael Seema, who will be telling us about things that are coming out of his laboratory that we think will represent very interesting prototypes, if you will, of MIT innovation heading towards the lives of patients. And then the third lecture we have in each of these series is really from the commercial voice, someone who has been involved in either running companies or building companies that are expressions of how innovation can, in fact, truly come from places like this into environments which can allow it to make its way into the lives of patients. And so we have another one of these programs in January. There'll be one in the early summer. Again, I do really hope that you'll consider coming to as many of them as you can. And I can't promise it, but I bet it's not going to be as ugly next time as it is this time outside. So with that said, we'll move into the introductions, and I'll tell you a little bit more about our speakers. After each speaker, we will break for a moment and entertain some questions. We do have these cameras rolling, and so that means we're videotaping this event tonight. It'll stream from our website over the next few days. It'll pop up online. If you do have a question, and we hope every one of you has a question, I'm not sure how we'll tend to all of that, but we do hope it's a very active, robust dialogue. If you put your hand up, we'd like you to do it into a microphone, so that your question can actually be heard in the recording, and therefore the people who are watching the video can have a chance to hear what their response is really all about. And we have people who'll be running up and down the aisles with microphones in their hands as we move into the question and answer session. And then after the 3 talks, we'll have additional desserts and coffee out there for those of you who are willing to stick around a little bit longer, network with each other. And we look for these events to essentially be accountable, if you will. We look for relationships that have emerged as a result of these programs and projects and other things that have started really as a result of us gathering on these types of occasions. So we'll start with Dr. Barbara Smith. It's important for me to be able to say it when I can that Barbara's one of our own. Right? She has come from MIT. She's currently the director of the breast program and co-director of the Gillette Center for Women's Cancers at MGH. She's an associate professor of surgery at Harvard Medical School and at MGH. Aside from having a degree from MIT, she went through the HST program to be an MD PhD at the end of it all. Her research activities have focused on reducing the extent of surgery and radiation required to treat breast cancer. She has a strong interest in new types of diagnostics and treatment modalities, and has spent a lot of time thinking about the role that 3-dimensional imaging and anatomy can be used to further reduce the amount of surgery required in treating particularly young women with breast cancer. So with no further questions, we'll have Dr. Smith join us.
Well, thank you very much. This is wonderful to be here, and my roots at MIT have really helped me and led me along the way. I'm glad to be part of this tonight. What I thought I would do would start by just a few slides to frame what are the current breast cancer problems that we have, what are we thinking about, what are the, in broad strokes, what are the treatments that we use, and what are the challenges that we face, and places where there's plenty of room to move things forward. So just to remind everyone, breast cancer is an extremely common disease. About 1 in 8 American women will be affected over their lifetime. That translates into almost 290,000 new breast cancers each year in the United States. It's a disease we do pretty well with, so the deaths are actually a small fraction of the cases. Most women will be cured and will not have a recurrence. But it's a disease where there's a lot of risk of getting new cancers. You get a cancer in one breast, that says you're at risk for new cancers throughout your breast tissue. So there are a lot of survivors who need to be followed, treated, and thought about. And those are the patients we're thinking about and working with. Over the last 20 years or so, the rates of breast cancer in this country have actually gone down a little bit, and deaths have also gone down a bit. And we think this is a combination of early detection and better treatments. However, around the world, rates of breast cancer are increasing. Countries in Asia that had traditionally very low risks of breast cancer are suddenly seeing a huge increase in their rates of breast cancer, the entire Asia-Pacific Rim. They think it's perhaps a westernization of lifestyle, other sorts of factors are playing into this. But what has been a huge problem in the United States and Europe is becoming an even greater global problem, and delivering care and managing it in different countries, different kind of healthcare settings is going to be an ongoing challenge, and it's 1 million new cases in the world at this time. So the way we think about treatment for breast cancer, and I'm using breast cancer as the example and as my own field, but the same issues and the same general treatment modalities apply to lung cancer, colon cancer, most of the common cancers that adults will get. And so we need to do local therapy, which is basically treatments in the area where the tumor arose, where you use these tools to first debulk the cancer, to remove as much of it as you can, and that's what surgery is still the best at. And then you use radiation, which you can aim to only a limited part of the body. You can't radiate the whole body without creating too much damage, but you can give a lot of radiation to a small area, and that can be very effective at killing small amounts of tumor that you can, if you can identify where they are, and can protect the other tissues. And then what kills people from cancers is usually not what's happening in the local area where the tumor arose, but in fact the mayhem created by the cells that travel to other parts of the body and grow in organs where you can't do surgery or you can't do radiation, and ultimately overwhelm the body's ability to function. So in addition to the local therapies, the surgery and radiation, we add medications that get into the bloodstream, can circulate anywhere, and can kill tumor cells wherever they might be. The issue with all of these things is that the difference between a normal cell and a cancer cell can be very subtle. And the treatments that can kill the cancer cells are often at great risk of harming normal tissues as well. So selectivity is what we look for. The challenges we have for the local therapies are that they hinge on the ability to know where the tumor is and where it isn't, to know the size and shape of the tumor. The breast has sort of very loose structure, so tumors can grow in all sorts of shapes, but we need to see where each of their little tentacles are in order to do the surgery. These things play into our treatments for our tools for treating the cancers, but also our ability to diagnose them. If we can figure out how to see them, we can use the same tools for early detection in mammogram innovations and other types of imaging studies. For the systemic therapies for breast cancer, there are drugs that both kill any fast-growing cells and others that really hone in on the estrogen axis of breast cancer. And we work at drugs that really are targeting it in those areas. So right now we have, for most women, have the option of a lumpectomy that removes only part of the breast, and instead of the old way of removing the whole breast. 20-year follow-up tells us that those two treatments are equivalent in terms of survival. And we now, by the way we do the lumpectomies, can make it so for most patients you're in about a 2% risk of a recurrence of that tumor in the breast after you've done the lumpectomy and radiation. The problems we have, though, is that we are still very limited in our ability to predict the tumor geometry before the surgery happens. We have some tools that help us, but in fact, a very high percentage of patients, after I do a first lumpectomy, I have to go back and do additional surgery and taking more tissue from one edge of the cavity I've created or another, just because at the time of surgery you couldn't see it. The pathologist can't give you the information in real time, and your imaging studies ahead of time didn't tell you what you needed to know. The strategies that we're kind of thinking of sort of in broad ways of how to avoid this problem of not getting the surgery right on the first try are listed here. Tools that would potentially give us improved preoperative imaging that would help us know exactly what tissue to take out. Am I taking something that's spherical or oval or with something going out this way? That would help. improved ways to image the specimens, which you can do things to a specimen you can't do to a person. So when I do a lumpectomy, I take the piece of tissue out and I can subject that to all sorts of testing once it's out of the person, as long as it is fast. The amount of time that it takes to do that's really critical for those sorts of tools. So if you have a tool that helps you do that, that would be fantastic. And the other thing is that maybe you don't image the specimen, but maybe you have something you can image the cavity that you've created that will show you in real time where the residual tumor is and let you excise it. So these are the wish lists I was told to come up with when I came here. So here's part one. A few things about what we can do now. Right now, a mammogram, which is basically an X-ray of the breast, produces the kind of picture that you see on the left side. The white is fibrous tissue. The black areas are fat. The younger you are, the more white tissue you have, and the harder it is to find a tumor as it's growing. What you see on the right side is a breast MRI. Gadolinium contrast is injected intravenously. It will highlight areas of blood vessels. Most breast cancers and many tumors of many types will have extra blood vessels that they're able to recruit in to feed themselves and help them grow. And the vessels are often abnormal and leaky, so the contrast is in a higher concentration because of the high vessel concentration, and it stays in the tissues because of the leakiness. So this has been a great tool for breast cancer. A mammogram costs about $200, an MRI costs about $3,000. So all of these tools that we are now also thinking about costs as well. A group at MGH recently developed a new kind of mammogram machine that basically, by having a X-ray source that moves and takes a series of pictures, in essence lets you look at individual slices of the breast without all the background fibrous tissue of the entire breast. And this just shows you that something on the left, you see this, and you may be able to see this sort of spiculated area here. That's a very small cancer that was almost invisible in the traditional mammogram, but when you could see sections of tissue in isolation, was easier to see, letting you find smaller and easier to treat cancers. Our group has been looking at different ways to image specimens. It turns out there are a number of small CT scans that can produce images in a rapid time sequence. Most tumors are denser to X-rays, so this is one tool we can use, and these are just some examples. If you take edges of tissue at the edge of a lumpectomy cavity where you can sometimes see residual material you couldn't see with the naked eye, and know that you needed to take more tissue in that area. The other thing our group does, because we're interested in anatomy, is we've really tried to look at what do tumors look like on a microscopic level as they grow. And breast cancers grow in many different patterns. Different subtypes grow with different patterns. And the strategy that you need to take out, this is a ductal cancer, which is about what 90% of breast cancers are. These tend to grow as fairly spherical objects that are pretty easy to get out. This is a lobular cancer, that these are both different views of the same tumor. And this is just a piece of it, but it grows with these very complex, invading small areas that are much harder to get around. So the tools you'd take for this are different than you might need for this. And this is a DCIS, or intraductal tumor, where it's almost this little cloud of areas that are involved, and some of these can actually extend at great distances beyond. We spend a lot of time creating these images from individual pathology sections at 50 microns and using a program to reconstruct them, because our feeling is if we don't know what the true size and shape is, we can't help the people that are developing imaging technologies and so on know what it is they're actually trying to see. So we're trying to have a sort of collaboration with imagers and the basically pathology things we can do, saying this is the true image, make what you show us ahead of time look like this. Just a few things about the drug development side of things and the systemic therapies. Many of the innovations in biology have really helped us understand how tumors grow, where are their vulnerable points, and try to exploit some of the subtle differences between normal and tumor cells. For the most part, clinical care now is based on things like this. So these are usually 5 micron thick sections of the tumor, fixed and stained, with a variety of things that help you highlight properties of the tumor. And this is basically all we've got right here for most patients. So this is the H&E, that's the classic way that pathologists tell one tumor type or one cell type from another. These are estrogen and progesterone receptors, are key things in regulating breast cancer growth, that there's immunohistochemistry and antibodies that help you see if these proteins are present or not, and present at high levels or low levels. And HER2, which is a surface protein that is overexpressed in some tumors and dictates how their behavior will be and what their treatment should be. But basically, we look at things microscopically with a few tools, and that's then what judgments are made on. The problem that this gives us is illustrated here, where we have two different patients with basically identical patterns of growth. The tumors were about the same size. There had been no spread to the lymph nodes. They both get tamoxifen, which is an estrogen blocking agent. And one patient has metastatic disease 3 years later, which is a fatal problem. And the other person is 12 years later and nothing has happened and probably never will. And our current tools didn't let us distinguish one of those patients from the other. So we either undertreated this person or overtreated this person with a lot of the options that we had. What's happened with some of the tools and understanding how genes are in chips, where you can test many different genes in the same small amount of tissue, has been we've recognized that there are many different genes that are relevant to breast cancer growth or to any tumor that you're looking at. And you can use these to get much finer granularity in understanding of the tumors and be able to categorize them in ways that can relate to drug sensitivity and other things. And so this has led to us rethinking how we classify breast cancers based on some of these gene profiling. And then lets us come back to these two patients again and try to understand what's different about them. So one company looked at patients like this who had what looked like favorable breast cancers, were treated with tamoxifen, and then looked at 10 years later and saw who did well and who did poorly, and then took archived tumor specimens and did gene expression profiling to find out what's different, how could we have predicted ahead of time which was which. And so this is the Oncotype, which is now a routine clinical tool that insurance pays for. It recognized that you could develop a pretty good algorithm to distinguish tamoxifen sensitive versus tamoxifen insensitive with a weighting of 21 different genes. And now I order a test. Some of the patient's paraffin-embedded tumor gets sent to this company, and I get back a printout like this that has a recurrence score. It tells me that this patient's risk of recurrence of her tumor as metastatic disease in the next 10 years is only 6% if she takes tamoxifen. That's pretty good. I see here she is on the graph. I might have another patient who would have a score here whose risk of tumor recurrence was 30% despite tamoxifen, and that's someone who should have chemotherapy who would benefit. And so this is something that for $3,500 the patient can have, and it has made it so that we give a lot less chemotherapy. So it's a way to not only, some of the tools we have us give more therapy. This is one where we actually give less, that's a very good thing. So the idea of this and the jargon we're using is targeted therapies. It's a way that you can potentially kill an individual's tumor better with fewer side effects and less toxicity. At the moment, there are a small number of tumors where this is applicable, but there are certain lung cancers, certain GI tumors where you can, if the tumor is of a certain specific type, there's a very targeted biologic often agent that will work as opposed to just a more generic chemical agent. There's a lot of potential targets. It's hard to figure out which the right ones are. It's hard to know which targets might be applicable to many tumors versus only one or two types. And there's a lot of this being looked at now, but it gets into the clinic pretty quickly if it works. So I'm stopping there and be happy to answer a few questions or save things till the end, whatever's best.
Questions now would be fine.
Hi, thank you for a great talk there. You started the presentation. Oh, sorry, my name is Ronnie Hansen, and I'm here from oncology cancer company called BioSeek Group. And I'm more interested in what you said in the beginning of your presentation regarding the screening which is going on, and you kind of alluded to that, the method of choice would be MRI for screening for breast cancer. But then, given the controversy of the current screening, would you say that MRI would actually, be so much better in detecting breast cancer in women?
So I think you're talking about the controversy with screening for this, and again, it applies to other diseases, is how hard do you look for something that's a relatively low frequency event? What kind of false positives and false negatives do you put up with, and how much are you willing to spend? So at the moment, the best tool we have, and there's a lot of debate about it, but as I read the literature, the mammography does certainly let you find cancers earlier. If you look at populations that are screened, the death rate is lower. It's a cost issue. Anyone who's had a couple of mammograms stops having those false positives. You kind of shake those out early in the process, and then after that, it's really not a lot of false alarms. MRI right now is so expensive that it has to be used very selectively, and the current recommendation is it's used only for women who have a very high risk of breast cancer, and it's probably 20 to 25% lifetime risk of breast cancer is where it's appropriate. We use it a lot in diagnostics. If someone has a cancer and we want to know more about its shape to do the surgery, that's another different issue. That's learning about a specific thing you're treating. But for screening, until it gets cheaper, it's not going to be the way to go. The thought with this tomosynthesis mammogram that was looking at slices, that's part of what's better about MRI is you're not looking at the whole breast at once, you're looking at sections. And so other technologies may get around that. Yes.
Going back to your comment about Asia, the fact that the incidence is rising, is that perhaps related to more intensive screening?
So this is the incidence in Asia going up is probably not screening because they do very little. And what we think it is, it goes back to some of what we understand as basic causes of breast cancer, which is longer exposure of the breast to cycling estrogens that happen with each menstrual cycle. And so the earlier a girl starts having periods, that adds to risk. The later she has her first baby, the greater the risk, because something happens when the breast differentiates in preparing to nurse a child that lowers its risk forever. And then the longer you continue to have periods or can take extra estrogens, the higher the risk. So what's happened in these Asian countries, the average age for women starting periods used to be 17 or 18, and now it's 13. And kids used to have, they'd start their periods and then they'd have a baby within a year or two, very young and start their families. And now periods start early, childbearing is late, few children are had, and that's probably accounts for a high percentage of the risk. And the reason kids are having their periods earlier is that nutrition is better in childhood. So you don't want to that's not something you want to go back to the bad old days for. What's actually of interest is people have thought about, since there's a very protective effect of a first birth, that to find something that can make your breasts think you were pregnant without actually having to have a teenage pregnancy. So if someone is working on that, that'll be useful too.
Question here.
The two questions, if I may. Firstly, during surgery, why can't you look at the resection margins by frozen section and figure out what how much you need to debulk that way? And second question, you mentioned that the death rates have been declining, and that's good. I read not so long ago that the mortality among men who are diagnosed with breast cancer is still about 75%. And why is that different?
So let me do the men first. The incidence of male breast cancer is about 1,500 a year in the United States compared with just under 300,000. And I think because of that, it's not very often found early. Men are not screened. Little lumps and bumps are often not dealt with early. And so it's kind of a delayed diagnosis. The stage for stage, so if you take a man and a woman who have the same size tumor and the same lymph node situation, they'll do about the same. But the men are rarely found when things are small. They're always palpable and bigger before you find them. So that's part of it. The treatments seem to be the same. The anti-estrogens work fabulously in men. And so that's probably what's going on there, late detection. As far as frozen sections, that has been tried in the past. The problem you have is that we care about every bit of the margin. For every bit of a sphere that's maybe 5 centimeters or greater in diameter has to be vetted by the pathologists, and they are looking at less than a centimeter square at a time, and the difference in some cases between the normal and the tumor is subtle. So it's unfortunately it doesn't work for breast cancer, though for some tumors like skin cancers and certain other things you can do it in real time. So we need something that gives us a bigger scale in a faster way. Mm-hmm.
The new technology, the
Tomosynthesis, yeah.
tomosynthesis,
Mhm.
how is that different than a CAT scan?
So this is something that's designed just to look at the breasts. It is a CAT scan actually uses contrast, and it is sort of a cross-sectional imaging. It's similar in that, computed tomography or tomosynthesis, same thing. This is actually using a technology that was used before CT scans were invented, which recognized that a moving source in essence has only a small area in focus at any one time, and you can kind of pull that out and look at it. So it's basically a moving X-ray source without contrast that gives you cross-sectional slices. So it's a cousin of it. Yeah. And just,
I have one last question that we should probably move on. How different is it in your work experience every day today than it was, say, 10 years ago? How quickly is the world changing for you?
There's very little that I do now that's the same as it was 10 years ago, and almost nothing from when I started my training. The idea of making the surgery smaller, we use radioactive agents and gamma probes intraoperatively to see where things are and take them out. The idea of targeted drugs, the idea that you're not just looking at the appearance of things, but coupled with that is actually a renewed recognition that certain anatomic structural things are actually very critical, that how cells spread is actually along specific structural elements that you can maybe manipulate or map and so on. So the ability to look inside the cells but also look at the anatomy in a different way has been really very helpful. And it's you have to enjoy doing new things every day, and luckily I still do. So.
I'll call it encouraging.
Yes.
Good.
Thank you.
Thank you. That was wonderful. We will have time at the end to ask additional questions, and obviously we'll have a networking opportunity for you to ask personally if you would choose to amongst the speakers. Our next speaker will be Professor Michael Sema. Michael is the co-professor of engineering at MIT and one of our intramural faculty members. That means he lives here. His lab is in this building.
Right upstairs.
Very important to me. One of our
Night and day, right upstairs.
One of our embedded guys. He was appointed the faculty director also of the MIT Lemelson program in 2009, which is an important nationwide program to increase inventiveness in our youth, something I know he's very passionate about, and you should keep your eye on that program. He brings remarkable groups of kids to campus to be involved in being very innovative. He's a co-author of over 200 peer-reviewed scientific publications, nearly 40 patents, as a recognized expert in the field of material science and biocompatibility. He's also a little bit entrepreneurial, to say it mildly. He's been involved in the founding of Microchips, Transform, more recently T2 Biosystems, Springleaf Therapeutics, and Taurus Biomedical. So he's taken things from the MIT laboratory time and time again and found a way to put them into environments where these can turn into products. And I suspect he's going to tell us about a few of those today. So we're going from the wish list to the prototyper, and we'll be looking forward to seeing more.
Well, thanks, Robert. Thanks for inviting me here tonight. I'm actually going to just talk about one project, but I am an engineer and a scientist that works on a variety of medical technologies. I also have the pleasure of being the teacher here at MIT, and I teach the first engineering class that students have an opportunity to take here at MIT. So these 400 freshmen arrive at MIT in my class, and they're very aware of science, what science is, but they have really no idea what engineering is. And so I have to tell them that what the difference is. And I tell them that great science is sort of at the boundary of what's known and what's unknown, but great engineering is at the boundary of what's possible and what's impossible. And of course, that's where we're at in cancer research, because a lot of the things we're asking were just a few years ago thought to be impossible. And so that's why it's a great field for engineers to work in. Now, clinicians have a great deal of advanced imaging tools like that shown here. And while these tools are impressive, they really do lack the chemical fidelity and specificity that are important to the biology of cancer. And, nothing beats the classic biopsy for diagnosis and staging of cancer. You don't get treated for cancer unless someone looks at a very small piece of tissue with under a microscope with immunohistochemical stains like were described in the last talk, and really characterize the cancer. And because this type of procedure is invasive, it's not the kind of thing you repeatedly do during the therapy phase. So while those exquisite chemical measurements are very important in staging and diagnosis, during the treatment phase, you lack that kind of information. So as an engineer, I see the tools that physicians use during the treatment phase as being kind of crude in many ways. And so the thought was, in this one project I'm telling you about, is what about if we take this invasive procedure and leave something behind that we can interrogate non-invasively, but still has that sort of chemical specificity, the exquisite characterization that you can get with a biopsy. And so the idea is essentially, can we take the laboratory and put it into the patient? So it's a little bit crazy idea, but you'll see in the progression of this how even very rapidly this has moved to extremely small scales. So it began by creating very small little hockey pucks like this that had the chemistry inside of them, immunohistochemistry inside of them. So instead of releasing the agent systemically, which has its own safety issues and that sort of thing, if you can make something, a little piece of plastic that you could leave behind during the biopsy and interrogate it, in this case with an MRI, you could then just know what the secreted factors are from the tumor or the tissue surrounding the device over a period of time during the therapy. And in this study this past year, we showed that we could get exquisite chemical sensitivity to nanomolar and picomolar concentrations of secreted proteins. In this case, it was a paper describing the markers for heart attack in an animal. And all the classic markers that are looked at in blood, we could detect by just imaging these very small pieces of plastic at some time after the heart attack. And so this sort of approach was something that we've worked on the past couple of years. And it was actually Herman Eisen, one of my colleagues here in the building, that a couple of years ago suggested to me, why don't you measure oxygen? And the reason why oxygen is important is that many cancers or many tumors are hypoxic. And it turns out this is integral to part of the biology of the tumor. And it's also clinically relevant. So because you can imagine, or many physicians can believe that the amount of radiation that you should dose depends on how hypoxic the tumor is. The theory is, and there's actually quite a bit of evidence for this, is that you need oxygen present to complete the damage induced by radiation. And in a hypoxic tumor, the necessary radiation has to be much higher. Of course, if you could measure the oxygen tension locally, you could adjust the radiation dose. but it's not quite that simple. There are contrast agents that purport to do oxygen measurements throughout the tissue, but because it's an agent that you're administering, how it compartmentalizes in the tissue varies from time to time. So these measurements don't give you an absolute measurement of oxygen tension. So what we did was we developed a material. So now you can see the scale. I'll back up here. So this scale here is about 4 millimeters or something like that. And now this material is a contrast agent in itself. It's about those marks are a millimeter in size, about 1.5 millimeter device. And you can see is that the measurement that you do in an MRI, the relaxivity, is a function of the oxygen tension surrounding this material. So this device very easily goes down during a biopsy. You can leave this behind, and of course then image it like is shown in the red arrow there. And just by measuring the contrast, you can tell what the local oxygen tension is. And so this is very recent, but what's really interesting about this is that I really do think that this, and we were already prototyping these, that at least an order of magnitude smaller devices can be made and imaged and accurately measure the oxygen tension. And so I hope over the next year you'll be hearing more about that. And so by starting relatively large and understanding the basic physics and what's necessary in your MRI to make these MR-sensitive materials on a large scale, we're able to begin to think to shrink these down. Now, I want to say something about MR, magnetic resonance imaging. I want to really thank the Koch Institute in particular, because in moving here, this really changed it's really changed my career in many ways, because I have access to a 7 Tesla small animal magnet right upstairs in this facility that I used to have to sweat blood to get access to just over a year ago. And it's really dramatically changed the rate of progress I can make. And so I really, having the state-of-the-art access to state-of-the-art research facilities is really has really changed my research. Now, we mentioned a lot about MRI, and it is expensive. Personally, as an engineer, I think there are opportunities to simplify these kinds of instruments, and certainly these companies are trying to do that. But is this concept? Can you take this concept to the next level? Do I actually need an MRI? And so I have students that are trying to replace the MRI by putting a little bit of the instrument. So we put the chemistry on the device. Can we put part of the instrument on the device? And so that's what this little guy is for. So you can see in this, there's a little coil here, and there's a little decoupling capacitor here. So this ends up being like a LC circuit if you're an electrical engineer. And inside here is one of these MR-sensitive materials. And what we can do with this is actually inductively couple and do the pulse sequence through this coil, but not with an MRI. We do it with a little handheld magnet and antenna. And so you can literally think of this as, we're not doing an image, we're just doing the pulse sequence on a very small volume of material in here. Now, of course, in my world, this is a very large object, but I believe the same sort of thing is going to happen as I build this at this scale, and I'm going to shrink this down to an order of magnitude smaller when I understand what's really necessary from the engineering point of view. So that means instead of an MRI, I can just take a little handheld device and stick it up to this and get the exquisite chemical measurement that I was just describing there in these other devices. And what that can lead to, I look at this, I think about a lot of these these types of potential products as being related to the world that we live in today. And the world we live in today is filled with screens. It's, I've got a screen here, I got one in my pocket, and all of you have one. And wouldn't it be cool if that screen had a check engine light on it? Right? I mean, I don't really need to know the ppm of troponin in my blood at any moment, but it'd be nice to know that you better check in with your doctor at the appropriate time. So I'll just end there and take any questions. Thanks.
Questions? I see hands up in the back. There's a microphone coming your way.
Thank you for your talk here. So I completely agree with you when you're saying that engineering is about dealing with known unknowns and science is dealing with unknown unknowns. So having said that, I think that I clearly see, from my perspective, a disconnect right now between engineering and science, and that is that engineering has caught up with medical science. You start off by quoting each other. I think there was, the scientists meeting up with engineers and, hey, could you help me out with this and solve this problem for me, and we started off from there together. And now the engineers has caught up with the medical science in that sense that when you look at diagnostic today, it is only as good as we are able to pinpoint, to tumor antigens and say that this is a positive answer in diagnostics. Otherwise, I've been looking at all of these nanotechnology and they are only as good as our biological understanding and being able to target. Otherwise, it's just another poison. Mm-hmm. So that's my question to you. Do you think there's a disconnect right now?
So if I understand your question is, do we lack sufficient basic biological knowledge to do appropriate diagnosis? What are the right markers for? Well, this is well, I think it's a real challenge. What are the appropriate secreted markers that you can look in blood to diagnose? They're very few. PSA, those are few, and even those are extremely controversial, to tell you the truth. And so I think that's it's a very serious problem. Now, what's happening there? You probably heard of circulating tumor cells, and now I think the whole field is switching to actually looking at small bits of DNA that circulate in your blood. And now, I doubt that kind of thing will be used for screening, but in the therapy portion of treatment of cancer, I do think these things are going to be important. We've gone through a huge wave of circulating tumor cell research, but I think now it's being followed by the realization that DNA in your blood does circulate. These are small segments. It gets chewed up a bit, but you can fish out a signal from a very low concentration in the plasma, and I think this is going to be a new wave of interest in not screening, but at least helping diagnose diagnose the progression of the disease, measure the progression of the disease during therapy. And that's a project, actually a different project. Love to tell you about some other time.
Very active area of research here, as you can imagine.
Yeah.
We have great tools. We need to have great analytes that can be used with statistical rigor to make these diagnoses real.
My question is, you've described the use of the approach that you've invented to monitor the percent oxygen in tissue. What about looking at other properties, biochemical functions of tumor samples versus normal, non-cancerous tissue samples? perhaps using magnetic resonance spectroscopy or some other approach like that, and seeing whether, just to give 3 quick examples, NADH/NAD plus ratios differ between the two, or intracellular pH differs between the two. Or finally, one can see differences which are known between cancer and non-cancer cells using MRS, whether one can see differences in the way that glycolysis is conducted in these two types of cells, because cancer cells, almost without exception, use an anaerobic form of glycolysis.
That's right. So I'll just comment on one of those. We have sensors that can measure local pH and response to therapy in tumors that can't be resected. It actually, you can see the change in pH with therapy. And it is, we are looking at, the easiest thing to describe here is that these sensors look at the tumor microenvironment. So any change in the tumor microenvironment, any of the secreted factors, we can measure locally before they get diluted in the blood. So if you take a blood sample, of course, you have no information about what's happening locally. But we can do that with this kind of an approach. But you're absolutely right. There are a lot of people in the building that study what's happening in the microenvironment of the tumor. Local cytokine concentrations, local markers of inflammation. As you know, there's a close relationship between tumor biology and inflammation. And these are the kind of things that we can measure. Unfortunately, the things that are clinically relevant right now are things like, pH may be one of them, but local oxygen tension is something that I actually get the biggest pull from this project from physicians on that. Can you tell me what the local oxygen tension is? And so Herman was right from the beginning.
He's always right.
He's great.
Scary right. Any other questions?
The key word in cancer diagnostics is specificity. So in the medical universe, a lot of researchers are looking at, as you mentioned, DNA. There's proteomics, there's metabolomics, there is transcriptomes. Which one of, so you cited the example of oxygen. Okay, that's what do you think is the next wave of cancer using your technology for cancer diagnostic? Which do you think is the next wave?
Well, I mean, these, I'm not a cancer biologist, so I'm learning a lot very rapidly, but I think what I have to do is follow what's relevant clinically right now. The problem with the diagnostic, any particular diagnostic or proposal of a diagnostic, is you have to prove that a physician can make a clinical decision based on it. And of course, that requires a lot of data. That's true for any chemical measurement or even the imaging measurement. So they're going to make a decision on whether or not to give somebody chemotherapy. It better be right. And so I get pulled in the direction. I work with a lot of clinicians, and I ask them how they spend their time during the day, and what keeps them up at night, and that's how I derive a lot of my projects. I think that's the nice thing about the Koch Institute is they've introduced me to many more physicians than I would have had at the other end of campus, and so I have to work from that. Yeah, I have to work from that end. We have a lot of people in the building that are on the other end trying to decide what are the right things to measure and trying to make a proposal that it's worth doing those clinical studies to prove that it's clinically relevant.
Perfect.
Okay,
We
thanks.
should probably move on. Again, there will be more questions at the end, and we'll have some networking, but we don't want, for those of you who want to leave on time, to be running too late in the weather and what have you. So it's my pleasure to introduce the third speaker. We've gone from unmet needs and wish list to prototyper to now, I guess we'll call it translator and or enabler. Ed Canna is the managing partner and chairman of Flagship Ventures, a firm he co-founded in 2000. Ed has over 27 years of experience in the venture capital industry. Prior to co-founding Flagship in 2000, he spent 14 years as general partner at One Liberty Ventures, predecessor firm of Morgan Holland Ventures. Ed earned his graduate degree, or his undergraduate degree in physics, Dartmouth, and went on to get an MBA, Harvard Business School. Ed has been a very active investor in our community. He started lots of companies that have brought programs forward, and I think he's going to share with us today some of the wisdom that he's gathered in helping to create those companies and some of the examples that have emerged from places like this into the lives of people.
Thank you. So, Robert, I appreciate not being described as I often am as a capitalist, because it would have worked the clinician, the inventor, and the capitalist. I love the concept of being an enabler. And indeed, one of the very special things about the Coke is that it combines the clinical folks who understand what the problems are, the people like Michael who intuitively know that if you take a rock and stand in a hospital and throw it in any direction, you can find several things that you can do better. And some collection of guys like me who are actually welcomed around here to wander around and ask questions, cheer, and occasionally provide some money. So as a enabler and a capitalist on this rainy night when Harvard is, I guess, on its second night of Occupy Harvard, how they can do that and not conclude that they are the 1% occupying themselves, I don't know. MIT is way too sensible for that. I thought I'd offer a brief perspective on the world. See if I can get this to change. This is actually apropos after a stock market day that went down 350, 360, then went up 3-something today. And the remarkable thing about this is that this is a 15-year-old cartoon. But I think it really does describe the world we live in. And when one thinks about translation, actually the context in the world are very important. Another just starting thought, really inspired by the fact that I'm following Michael, is for the innovators here. And this is, I believe, an Einstein quote, and I think it so well describes what goes on in the halls here. If the idea is not absurd at first, then there's no hope for it. Going into healthcare, I have a minor addendum, which I'm going to add here, because in today's world, it's not just enough to come up with better ideas that improve outcomes. You've got to also lower costs. So that's pretty serious today, a real challenge for all innovation here. It not only has to improve quality, it has to lower costs. Okay, why is the Coke important? Why is cancer such a significant disease? This is a fairly new look at world cancer, similar to Barbara's numbers from the WHO. And what you find if you look across the world right now is you find darn near 15 million people getting cancer every year. A large portion of them, and you can see this arrayed by cancer up here, a large portion of them are lung, so 1.6 million lung. The red is the incidence per year, the blue is the mortality. Pretty startling figures. If you take it down to the US, it's approximately 1.5 million people per year getting cancer. The average cost of treating that is about $90 billion direct, about $120 billion indirect, huge numbers. And the thing that's even more interesting in some ways is that on top of this incidence number, the new cancers diagnosed, there's what Barbara described, which is the prevalence number. Depending on where you are in the world and how quickly you die, the prevalence could be 5 to 8 times the incidence. So we're looking at a prevalence in the U.S. that is a multiple, is about 11 or 12 million people who've had cancer and who are at the risks that Barbara described of recurrence, and a multiple of that in the world. So it's a big problem. So here is a look at the venture capital involvement facing that big problem, and it's kind of humbling because the dollars involved aren't large. This is venture investments in cancer from 1985. I couldn't go back to 1983, which is when I got into the venture business, but 1985, up through 2011 year to date. Recently, we've been pumping as a venture industry somewhere between $1.5 and $2 billion into the cancer research ecosystem. Not a lot of dollars compared to pharma dollars, government dollars, other dollars, but it is precious dollars because it occurs at the interface of the innovators and the clinicians. Just focusing in on the last 10 years, so 1995 to 2011, and moving from cancer to all of life science investing. Here you see sort of the industry at large going from about just under $2 billion in 1995 to several years here, where we've been in the in the kind of the $6 to $7 billion range, pumping into solving healthcare problems. You see the split between devices, services, and biotech drugs. And typically devices are about 35% of the overall investment that guys like me make in the system. Here's a look at the direct cancer investments in the VC community over the past 10 years. You can see it spiked up considerably in and around the 2000 timeframe with the sequencing of the human genome, which really did set a new bar for innovation in the sector, as has been described. And again, it's been pretty consistent at about 25% of all VC dollars go towards solving this set of problems in the cancer area, representing $90 billion of spend and cost in the US economy. Okay, and finally then, to take it one cut further, and I don't expect you to see too much here, but here's a look in the cancer sector of how you divide the VC expenditures between diagnostics, devices, tools, and basically biopharmaceuticals, and plus or minus 40% goes into non-therapeutic type things, the sort of things we're talking about tonight, and that's split about a third into tools, a third into devices, and a third into diagnostics. Okay, so that's the lay of the venture capital land. So in preparing this talk, I find myself asking sort of a fundamental question because actually much of the excitement in and around Boston relating to cancer developments is pharmaceutically, biologically related. And I found myself wondering what a cancer device was really. And it rapidly came to me that the answer was in fact embedded in this graph. Those of you who've been around will recognize that this is the org chart for the FDA. And these are the three key branches of the FDA. And so the answer as to what a cancer device is it's anything the FDA wants it to be. And typically the Center for Devices and Radiological Health is a key approver in the regulatory process that the such devices go through. Now, since the government is good at spoon feeding us, they actually gave on their FDA website and regulations a very complete definition of what a device is. A device ranges from tongue depressors to programmable pacemakers to in vitro diagnostics, to general purpose lab equipment to electronic radiation emitting devices. And if you're not quite sure if you fall into that, they've got this marvelous new creation going back to 2002 called the Office of Combination Products, which is intended to arbitrate a lot of what exists today, which actually exists at the interfaces of all the above. Okay, so that's, with that as a background, let me talk briefly about Flagships' involvement in cancer devices. And it really goes back to my entry into the business in 1983. Again, most of what we do is related to the therapeutic side of things, but actually, as I looked, we've been involved a lot in devices. When I got into the business, looked around, there really wasn't that much to do in cancer, improving cancer, except for supportive care called palliative care, imaging, which was really expensive and hard to make a dollar out of as a small company, and novel diagnostics. So one of the first things that we did was to come up with a sort of an improved way of controlling cancer pain. And this is a lollipop-like device that causes a potent morphine analog to rip across the mucous membrane of the mouth, gives you a jolt of pain relief to control your cancer pain if you're dying of cancer, doesn't give you a rush, And it actually ended up being about a $500 million per year product over several years and really has changed the end stage life of many people with cancer. 1987, we were the founders of a company called Cytic, which observed that for frequently screened patients, cervical cancer was a non-issue. In 1986 and 87, commerce got the better of pap smear screening, and the idea in pap smear screening was to underprice the competition and have cytotechnologists sort of wave their hand over the slide and say, did it have cancer or not? And we set out to develop a robot to read the slides. We ended up discovering that the problem was we actually needed to come up with a better preparation. So ThinPrep, pioneered by Cytic, now is the dominant mode for pap smear reading. It's about 80% of all paps in the country are done through Cytic, a local company. And these are long-term endeavors, guys. So the process of translation to significant impact in medical practice is a long one. Exact Sciences, where we were founding investors, we tried to take the ideas of Cytic for cervical cancer screening and say, why not do better for something in colorectal cancer? And two preeminent scientists, Bert Vogelstein at Johns Hopkins and David Ahlquist at the Mayo, had thought deeply about colorectal cancer, realized that the DNA from the cells, cancerous cells, was shed in unusual proportion in the stool. And we had the conceit to try to build a company to try to do stool DNA testing to try to come up with a non-invasive screen for colorectal cancer. It's been a long time coming. Recently presented some results at the AACR showing 87% sensitivity, about the same specificity, so pretty extraordinary results that now need to be validated in a large-scale clinical trial ongoing. So in 2000, we shifted gears, and this is where Flagship kicks in. Got involved in biomarker-based diagnostics, got involved in single-molecule DNA sequencing, particularly focused on cancer, got involved in molecular imaging, got involved in nanoparticle drug delivery. Again, one of those convergence technologies. Got involved in real-time visualization, something that Barbara spoke a lot about, and a company that Michael didn't talk about, but has huge implications in bladder cancer. Got involved in a company called Taurus, which tries to take the existing drugs used in bladder cancer treatment and make them that much more effective by effective drug delivery. So that's the Flagship involvement in bladder cancer, in cancer devices. During the time that I've been in the industry, I've had the privilege of seeing cancer treatment just dramatically changed. And it was a question you asked, Barbara, but it's just remarkable. Before tonight, I actually attributed most of that to a little bit of imaging and a lot of biotherapeutics. But in thinking about it, it's much broader and deeper than that. And the device guys have had a huge, huge impact on cancer therapy. So digital imaging technologies, they've been mentioned, but CT, ultrasound, the whole concept of image-guided biopsy, taking PET and combining it with CT and MRI, SPECT, in this emerging field of molecular imaging are really changing our ability to do early detection, diagnosis and staging, and then the follow-up and monitoring for this large group of patients who've had cancer in the past. There's this whole set of interesting eclectic technologies that have been brought to bear for novel ideas in screening, lights that go over the skin to pick up cancer, better ways of doing biopsy, such as Michael described and lots of people have worked at, and approaches in the molecular pathology area. Likewise, a lot of work going on in the molecular diagnostics and biomarker area, which is impacting how cancer is treated today. The three mainstays of surgery, radiation, and chemotherapy have had their own revolutions during the past 2 decades. A lot of it is device-driven. Radiation is robotically controlled. Chemotherapy is locally delivered and computer-controlled. And the surgical tools are getting better all the time. Biologics, I think, speak for themselves. And there's been a huge amount of device activity in a field called interventional oncology, which is trying to provide a set of tools for surgeons like Barbara to do their job better. So if you just think about the field of cardiology, it used to be a surgical field, now it's largely an interventional field, a question that some people ask, and in fact, Smart people will prognosticate. Will cardiology serve as a precursor for what oncology does, and more minimally invasive surgery become the mode of the day? And there's just a huge number of devices that have been invented, a lot of them here locally, either applying thermal techniques, arterial access techniques, or increasingly electrical techniques to trying to hit the solid tumors that are operated upon. It's an important and involving field here. It's another mantra that I've lived through for a decade now, called personalized medicine. It was first hyped in the press in about 2000. We're still not there yet, but we're getting awfully close. You don't have to focus on these things, but they're sort of lists of, they're sort of references of paired therapies and diagnostics. And more and more they're coming about. The pharma industry has paid it lip service for years because it cuts down their market. By and large, if you can, if your drugs only work for 30 or 40% of the people, your market is 2.5 times larger than it should be. But that is changing, and increasingly we're pairing biomarkers with pharmaceuticals, and the cost of developing these drugs these days is actually requiring it. So just as a parting thought, and this is intended to try to tie into some of what Barbara said and some of what Michael said, when I look across the nation, but largely the Boston community, there are a bunch of really extraordinary, important things happening. Some of them are commercial potential in the near term. Some of them will take a little bit longer to get there. But let me just describe briefly a few of them. This concept of healthy living, which Barbara talked about, is critical to reducing the burden of cancer here. I don't know how you build a business on it. I really don't. But I've actually gotten quite intrigued lately watching my 15-year-old talking to her iPhone 4S, that just maybe some combination of avatars and personal assistants, and there's some marvelous work being done here at MIT in the Media Lab focused on that. Just maybe there's a way that the buddies that we have in the computer world can help us live a better life. Don't know how to build a business there, but boy, if you could do one thing to knock down cancer, that would be it. Obesity, smoking, important problems. The whole field of diagnostic biomarkers, it's been discussed a bit here, but it is beginning to revolutionize the diagnosis, staging, and treatment of cancer. There's a huge amount of commercial activity ongoing right now. Lots of companies. Michael mentioned circulating tumor cells. Some spectacular work done here at MIT, a couple other groups around the country, finding nanotube chips to capture these circulating tumor cells, which enable you to get a visibility on what's going on in metastatic cancer in a really remarkable way. 3D imaging, I think we're at the dawn of 3D imaging. Barbara showed some of the recent developments. I think there's a lot more coming there. You're either a believer in imaging or not. I happen to rely on Star Trek for a lot of my future prognostications, and I do believe that we will figure out how to use imaging cost effectively, that it makes a difference, and over the next 2 decades, we're going to be able to do things both with digital imaging of various sorts and molecular imaging that will make the lives of clinicians, the lives of patients so much better. There's some very interesting work being done, again, here in and around MIT. Folks like Michael, Ralph Weissleiter over at MGH, trying to enable the real-time pathology bedside that Barbara's looking for, taking MRI, miniaturizing it, and allowing you to determine pathology at the bedside real-time. Advanced surgical tools, both doing conventional surgery and surgery via interventional oncology and stereotactic radiosurgery. Really local therapy designed to minimize the damage caused here. One of the privileges in my business is to be able to see ideas that I thought were Albert Einstein dumb turn into great ideas, and I was reminded of that tonight. There's a guy in the audience named Mike Ambrogi who 5 years ago joined a company called Novacure, which had sort of a sordid background out of Israel, got FDA approval in April this year to sort of beat down cancer of the brain by pulsing low field electrical impulses into the brain over a long period of time. Just a marvelous example of an inventor coming up with new ways to actually treat the tumors that are there. And then finally, these two areas of molecularly driven drug design. Can we come up with better ways more efficient ways to develop drugs? And can we actually take the drugs and make them tailored for the people the way they need them? They're big areas. There's a lot of money going into them. There's a lot of opportunity, and I'm a believer. So that, my friends, is sort of all I wanted to say. We're past our time. And there's a lot of work to do. There's a lot of interesting excitement out there, and let's go make it happen. So thank you.
Perfect. Questions? You know what?
And I'll join you for a drink is a fine question.
Right. Well, there's one right behind you first. Just 1 second.
What is molecular imaging?
So molecular imaging is a set of techniques that are designed to actually allow via imaging technology, typically probably the predominant one is fluorescence, but people would say that MRI and PET are also somewhat molecular, to actually look at what's going on at a molecular level. So you're looking at chemistry through an imaging technology, not just structure. And it's an alternate approach to achieving some of the things that Michael talked about. One of the great things in the field is that the most exciting thing, which is really where Barbara comes in, is how you put it all together. But molecular imaging is about a 15-year-old technology that's now made it into animal modeling, is beginning to find its way towards early human clinical application, and may allow you to actually see what's going on in a tumor in your body so you know how to treat it.
Any others? Right here, yes.
Do you think that we are doing enough from a regulatory point of view to support the development of personalized medicine? I'm thinking that we have fast track designation, we have orphan drug status. Is there anything else we should be doing from a regular point of view to ensure that the venture capital is being poured into this area and the companies really developing true personalized medicines have a chance out there?
Sure. It's a good question. It's kind of multifaceted. So there's a backdrop to the question. Life science venture capital is a bit under attack these days because as a financial investment proposition, most of the companies we back take a long time to grow and exit. And the returns haven't been as attractive as that which people are achieving by investing in Facebook and Groupon and a set of things like that. So capital tends to flow where money is made, not where need exists. One of the problems of capitalism. One of the reasons that occurs is, in fact, the FDA. And the FDA has a horrendously tough job, in part because in our society we have no appreciation of risk. And the FDA is held up as a regulatory body to keep us from being harmed by innovation. So actually, the first thing I would do if I could do anything is have people like Michael teach the entire country how to think about innovation and risk. It's a real tough problem. One of the issues at the FDA right now is that they are living through the tragic consequences of something called the metal-on-metal hip implant, which by all rights should have been an important incremental innovation over metal over plastic implants. A regulatory process was run that involved studies, multi-year studies, similar to that which had been done in the past. And unfortunately, 7 or 10 years later, once this technology is broadly deployed, we find out that it has some tragic consequences for a portion of the people that have the metal implants. What that's done at the FDA is it's opened up a free rein for any scientific question anyone wants to ask about innovation on the way to approving it, which is extending cycles a lot longer. And that includes diagnostics. And the diagnostic pathway, the FDA has gotten involved in a big way. So on the one hand, there's a lot of talk and a lot of enthusiasm throughout the industry and regulatory bodies about personalized medicine. On the other hand, when you're on the ground with a product trying to go through the FDA, funding a company, it's a daunting prospect. I don't know how to solve the problem. It's one of 3 or 4 systemic problems in the U.S. right now that are very daunting.
No, there's one more question in the back. You have the microphone now? Yes, go ahead.
I have a general question about nanoparticle drug delivery. I saw that you guys are an investor in Bind Biosciences, and I just want to know about your perspective and the challenges that your company sees in this field, in particular to do with how do you select the companies which you think will be winners in the huge sea of nanoparticle technology that's out there.
Sure, so that is the art and science of venture capital, and I'll tell you what I've learned after 27 years of doing it, which is I'd rather be lucky than smart. It really is, it's remarkable. That having been said, the analysis we do when we invest in a company is an analysis of potential. And the potential at the end of the day is determined by the people. And one of the privileges I have being resident in this region, working with people like those here at the Koch, is working with some of the best people who actually, when they run into the inevitable obstacles that pioneers always run into, they find ways around them in an event. And in fact, the odd thing about my profession is the value that is created in the companies I back is not actually created by what is done before I invest. It occurs once the play is started, the unanticipated occurs, and we run into the obstacle that we can't overcome, and a group of people overcome it. And that's where the innovation occurs. So in short, there's a whole range of new technologies that offer the potential to go from about 0.5% of the drug delivered systemically goes to the tumor, or some just absurdly low figure. Maybe it's 1%, but it's not much more than that. It's a little bit scary, to not 10, but 2, 3, 4, 5%. But that 2, 3, 4, 5% is a multiple, and the potential for these third generation drug delivery, largely driven by nanotechnology to impact cancer therapy, is huge. So we've made a couple of bets there. We've done it very selectively. We happen to have done it with people in and around the neighborhood here. We're feeling pretty good about those bets right now, but we've got a long road to go. Just started in clinical trials, and we'll see what comes of it. It's a tough job to try to be the arbiter of who gets money, because there are a lot of worthy people out there, and you can only do so much.
Okay. Any others? One last? No? Any other questions for the previous speakers? Okay. Well, I will then wrap us up by reminding you of a few things. I hope all of you have found this as remarkably encouraging and as inspiring as I have. Thank you for putting it all together and bringing us up to speed on the way in which you see the world. The next event that will be similar to this is in January. You'll see on your program an event. I don't have it memorized what date it is, maybe the 19th, something like that. Don't quote me. It's going to be about cancer plasticity, the role in which sort of stem cell-like biology plays in enabling or participating at least in the process of metastasis. We have some remarkable people speaking, not the least of which is Tyler Jacks, who's the faculty director of this Koch Institute. So he's as gifted as any amongst us, and I think he'll share with you, along with some of the other speakers that will be flanking him, some of the things that are really emerging in that space that I think could have remarkable translational potential. So I hope you'll include that. In March, we'll be back again, not for an event like this, because it'll be about celebrating the participants of our community in their efforts to create the images that are on our walls. And so we have an image award competition, and that's how all the stuff that you may have seen as you walk through the gallery, how that was selected. Well, there have been 10 new winners selected, and those will go up in March as we celebrate our 1-year anniversary of moving into the building. And I hope you'll think about coming to that event as well. And the young trainees, in most cases, will be talking a little bit about what he or she was doing when they took that picture, and what that image both meant to them aesthetically, as well as what the science behind it really means. And so I think you'll find those events interesting as well. So again, thank you all for coming. There is coffee, dessert, all the things you might want before you get back out in the weather. And we have really enjoyed your time with us.